In this lecture, we're going to be looking at the relationship between the behavioural changes observed during motor learning and the corresponding changes in functional neuroanatomy. Much of the material from this lecture can be found in Chapter 4 of the Shumway-Cook and Willicott textbook. So, in this lecture, we'll be looking at the evidence revealing the types of changes in the functional organisation of the central nervous system that go along with the behavioural changes associated with motor learning. Let's start by looking at a study by Asanuma and Keller. The hypothesis in this research is that learning a skilled action involves the long-term potentiation of specific cells in the motor cortex by cells in the somatosensory cortex. In other words, we're saying that learning is associated with changes to sensory-motor cortical pathways. Long-term potentiation refers to the persistent strengthening of synapses based upon recent patterns of activity. The researchers studied cats learning a new skill. Here we see a cat from the study reaching through a gap in a box, with the goal of obtaining the food pellet that's sitting in the beaker. A key part of the skill to be learned here is that there is a gap between the beaker and the box. Here we can see the significance of the gap coming into play. The cat reaches for the food pellet, pulls the pellet out of the beaker, and then the food pellet drops behind the box and out of reach. The skill to be learned here is how to get the food pellet without it dropping through the gap. Here we see the results of having learned the skill. The cat is able to get the food pellet because it's learning to supinate its wrist. If the cat learns to supinate its wrist, and therefore effectively cup the ball, it's able to get the food pellet into the box. So, we just saw the behavioural changes that were associated with the cat learning to get the food pellet from the beaker. What we're interested in is the corresponding neuroplastic changes. To investigate this, the researchers surgically removed the somatosensory cortex in one hemisphere before the cats had any practice at the task. An important detail of this research is that during practice the cat attempts to perform the task with both limbs. This means that we have an impaired limb that will be affected by the removal of the somatosensory cortex from the contralateral hemisphere and an unimpaired limb. Remember, our hypothesis here is that learning a skilled action involves long-term potentiation of specific cells in the motor cortex by specific cells in the somatosensory cortex. The researchers consequently predict that if the somatosensory cortex is removed, then the cat will not be able to learn the novel movement pattern. So what were the results from this study? After the practice trials, we see that for the unimpaired limb, the cat is learning the new skill of supinating its wrist in order to get the food pellet. In contrast, this skill was not learned on the unimpaired side. The conclusion here is that the somatosensory cortex appears to play an important role in the learning of motor skills, through the mechanism of long-term potentiation. We're not done with the research quite yet. The authors performed a post-test. They took the cats that we just looked at, that had learned the wrist supination skill with their unaffected limb, and they then surgically removed the remaining somatosensory cortex. So now the somatosensory cortex has been removed from both hemispheres. The results here are interesting and surprising. The removal of the somatosensory cortex did not affect the skill that was learned by the unaffected limb. In summary, the first part of this research is telling us that removing the somatosensory cortex stops the wrist supination skill from being learned. The second part of this research is now showing us that once the skill has been learned, removing the somatosensory cortex does not impair the ability to perform that skill. What can we conclude from this? In simple terms, we're seeing that the somatosensory cortex is necessary for learning a motor skill, but it is not necessary for performing an already learned motor skill. The reason for this is that the learning process doesn't only change sensoromotor cortical pathways between the sensory-motor cortex and the motor cortex. It also affects other pathways, such as the thalamocortical pathways. The implication here is that pathways are co-activated and long-term potentiated during the skill learning process, and even if one of these pathways is impaired, others are retained. So, we have the idea that during the motor learning process, parallel pathways are becoming long-term potentiated. Building on this idea, Guggenmuss and colleagues tested the hypothesis that recovery after a brain injury can be supported by facilitating new functional pathways between distant locations in the cerebral cortex. To test this hypothesis, the researchers injured the primary motor areas in test rats. This lesion disrupted the communication between the somatosensory area, S1, and the premotor area, PM. The lesion impaired the rat's reaching and grasping abilities. The idea at the heart of this research is that recovery after a brain injury can be supported by facilitating new functional pathways. To test this idea, the researchers attempted to promote recovery of function in the lesioned rats. They attempted to do this by creating a new pathway between the somatosensory area and the premotor area. To do this, the researchers created what they called a neural prosthetic by inserting microelectrodes into the cerebral cortex. This neural prosthetic uses one microelectrode that's implanted in the premotor cortex and another microelectrode that's implanted in the somatosensory cortex. The signal from the microelectrode implanted in the premotor cortex is analysed to detect action potentials. This measure of premotor cortex activity is then used to trigger the electrical stimulation of the somatosensory cortex. What we've done here in creating this linkage is connect the premotor cortex activity to somatosensory cortex activity. Now that we understand the design of this intervention, let's look at the outcome. When the impaired rats received the spike-triggered stimulation, reaching and grasping function was substantially improved within 1 week, and by 2 weeks it was indistinguishable from the pre-lesion levels. This research suggests that the neuroprosthesis created a new functional connectivity between the two target areas. The next piece of research I would like us to look at is by Floyer, Lee and Matthews. They hypothesized that the stages of motor learning are associated with corresponding functional changes in the brain. To test this hypothesis, they set up a motor learning experiment that could be performed inside an MRI scanner. While laying inside the scanner, participants held a force sensor. By squeezing the force sensor, they controlled the height of a blue bar that was displayed on a screen in front of them. The task for participants was to adjust the height of the blue bar to match the changing height of a red bar that was displayed beside it. The red bar had a complex pattern of motion, so the task here is to learn this complex pattern of force production, such that your blue bar will track the trajectory that we can see of the red bar height changes displayed on the right. So here we see some results from this experiment. On the x-axis, we have the different blocks of training trials. As we move from block 1 to block 10, we can see the changes associated with the learning that's occurring in this task. The line with the black diamonds shows the tracking error. That is, how accurately the participant managed to keep the blue bar exactly aligned with the red bar in the task. We can clearly see that the tracking error decreases over the training. The other lines show the force produced by the participants. These lines aren't relevant for our current purposes. The graph that we just looked at showed a clear reduction in targeting error over the training, suggesting that participants learned to do the task. Now let's look at the changes in cortical activity that accompanied the skill learning. Here we see the results of an fMRI analysis. The areas that are shaded dark blue identify cortical areas that show a decrease in activity with skill learning, whereas the areas in green identify cortical areas that show an increase in activity with skill learning. If we focus in on some of the specific cortical regions that are highlighted here, we can see the changes in activity over learning. For example, here we see the level of activity in the dorsolateral prefrontal cortex. In this graph, we have training block on the x-axis. We can see that the level of activity reduces over the blocks of training trials. Here we see the graph for the level of activity of the primary motor cortex. Again, we see that the level of activity reduces over the blocks of training trials. In general, what we're observing here is a progressive decrease in cortical neural activity as task performance improves. If you remember from a couple of slides back, one of the goals of this research was to relate the stages of motor learning to corresponding functional changes in the brain. So let's relate the findings that we've been discussing to the idea that there are distinct stages of motor learning. In the initial or early stages of motor learning, performing the movement is believed to demand more attention. In the later stages of motor learning, the movement is performed more automatically, and much less attention needs to be directed to the action. Floyerly and Matthews proposed that in the initial stage of motor learning, performing that action will be associated with widely distributed neural activity. They proposed that activity will be predominantly in cortical areas. Consistent with this, they observed significant activity in areas such as the prefrontal cortex, the sensory motor cortex, and the parietal cortex. In the later stages of motor learning, Floyerly and Matthews propose that performing an action will be associated with decreases in cortical involvement. Consistent with this, they observed a decrease in primary motor cortex activity and an increase in activity in subcortical motor regions. This included the thalamus, the cerebellodentate nucleus, and the putamen. The authors suggest that performance gains in the initial stages of motor learning strongly rely on interactions between the prefrontal areas of the cortex and the caudate nucleus, whereas improvements during the later stages of motor learning involve increased activity in subcortical circuits involving the thalamus, the cerebellar nuclei, and the basal ganglia. The broad strokes takeaway message from this research is as follows. Motor learning skill is associated with reductions in cortical activity in regions responsible for executive functions, processing somatosensory feedback, and motor planning. On this slide and the next, we'll be looking at research by Wu and Hallett. This research looks at the functional neural changes associated with learning a task to the point that it becomes automatic. On this first slide, we'll be looking at these changes in young and old adults. On the next slide, we'll also be looking at patients affected by Parkinson's disease. Wu and Hallett studied the task of learning a sequence of finger taps. For this task, you tap your right fingers on a surface placed underneath your hand. The simple version of this task requires you to learn to perform a sequence of 4 taps. The harder version of the task requires you to learn to perform a sequence of 12 taps. For example, in the simple task, you might be asked to perform the sequence of tapping your ring finger, then your little finger, then your index finger, and then your middle finger. Participants in this experiment were also asked to perform a secondary task at the same time as performing the primary task. This task involved watching letters flash up on a display screen and counting the number of times that a specific letter came up. For example, you might have to count the number of times that the letter G flashed up onto the screen in front of you. In this research, we're interested in the ability to perform movements automatically. Automatic movements are performed without attention being clearly directed towards the details of the movement. For example, musicians can perform music accurately while holding a conversation at the same time. A key characteristic of an automatic movement is the ability to reliably perform a primary task while also being able to pay attention to another secondary task. In order to get to the point where participants were performing the primary task automatically, the researchers had the participants practice until the primary task could be completed without error from memory, and could be done so at a speed of at least 2 taps per second, and while the secondary task was also being performed at the same time without error. Let's look at the results of this research. After practicing the task, both the healthy younger adults and the healthy older adults achieved automaticity. As you might have guessed, the older adults required significantly more training to achieve automaticity. Functional MRI scans were made both before and after practice. For both the older and younger adults, there was less activity after training. The image here shows some of the areas of the brain that were more activated before training than after training for the older adults. In the image here, the areas in red now show some of the regions that were found to be more activated in older subjects compared to younger subjects during automatic task execution. These results led the researchers to conclude that for older adults, obtaining the level of automaticity was associated with a greater amount of brain activity. The sharp contrast that we're seeing between younger and older adults here appears to be the main reason why older adults have more difficulty in achieving automaticity. We just looked at age-related changes associated with learning a task to the point that it became automatic. Now let's look at a second study by Wu and Hallett that studied these changes in patients with Parkinson's disease. This research uses the same design as the one that we just looked at. Participants again practice until they can get to the point where the primary and secondary tasks can be repeatedly performed at the same time without errors. So what did Wu and Hallett find in this experiment? Both the group of Parkinson's patients and the group of age-matched healthy older adults were able to achieve automaticity in the simple task. As in the study we looked at on the previous slide, all of the age-matched healthy older adults were able to achieve automaticity in the more complex task. In contrast, some of the Parkinson's disease patients were unable to achieve automaticity when the task was harder. What we're learning here is that some Parkinson's patients will find it very challenging to learn to perform a more challenging task automatically. For the age-matched control subjects, reduced brain activity was again seen when they were performing the task automatically. In contrast, the participants in the Parkinson's disease group showed much greater brain activity compared to the control group during the automatic stage. Here we see some of the brain areas that were more activated in the Parkinson disease group than the control group. From these findings, Wu and Hallett suggest that in order for people with Parkinson's disease to perform movements automatically, more brain areas need to be recruited to compensate for the basal ganglia dysfunction. In the last part of this lecture, I want us to look at a study by Jenkins and colleagues. This research looks at how motor learning affects the organization of somatotopic maps. Specifically, we're going to be looking at the mapping between areas of the body and activation of regions within the primary somatosensory cortex. Jenkins and colleagues did their research with adult owl monkeys. The adult owl monkeys were trained to reach through an opening in their cage to touch and hold the edge of a spinning disc. If the monkeys effectively stopped the disc from moving for 10 seconds or more, then a food pellet was released into the cage. The important detail of this training is that the disc was arranged so that only the middle 3 out of the monkey's 5 fingers could reach the disc. The monkeys were trained for approximately 10 days, and on each day they managed to stop the disc and get the food pellet on average about 600 times. Here we see the somatotopic cortical map of the owl monkeys before they began the training. Neurons in the area labelled 1 are activated when the thumb receives cutaneous stimulation. Areas 2, 3, and 4 are the areas associated with cutaneous stimulation of the middle fingers that we identified on the previous slide, and area 5 is the area associated with the little finger. Here we see the somatotopic cortical map after training. You'll notice that areas 1 and 5 are approximately the same size both before and after training. In contrast, areas 2, 3, and 4 have enlarged substantially. What we're seeing here is an enlargement of the cortical representation of those areas of the hand that are most frequently behaviorally engaged. We just looked at how the experience of repeatedly touching and holding the disc changed the areas that represent the fingers within the primary somatosensory cortex. Experience also affects cutaneous receptive fields. Cutaneous receptive fields are areas on the skin that, when stimulated, cause the same part of the primary somatosensory cortex to be activated. Here we see a diagram of the cutaneous receptive fields in the fingers before training. Each receptive field is outlined with a black line. If the skin is stimulated anywhere within a particular receptive field, then the same region within the primary somatosensory cortex will be activated. Here we see a diagram of the cutaneous receptive fields in the fingers after training. If you look back and forth between these two receptive field maps, you'll notice some significant reorganizations that are occurring in the receptive fields. Look specifically at the middle 3 fingers, labelled 2, 3, and 4. These are the fingers that the monkeys are using to stop the disc from moving. More precisely, it was the tips of these 3 fingers that are making contact with the disc. So what are we seeing here? We're seeing that with repeated use, the cutaneous receptive fields that are recorded within the cortical representation zones are becoming smaller. Another way of thinking about this is that the areas are becoming more focused. The fields are also becoming more frequently located at the tips of the fingers. In other words, they are in the functionally relevant area. In this short lecture, we briefly looked at the relationship between the behavioural changes observed in motor learning and the corresponding changes in functional neuroanatomy.